Hello everyone, my name is Tahir Shad and I am here to present part of the project I was working on over at NIAB as part of the Rokos Internship. So the work revolved around investigating nitrogen response in wheat. So to give some background as to why this is so important is because wheat is an incredible crop in of itself. It's most likely in everyone's diet, and so it feeds a lot of people, and it is also sold by people for a living. Nitrogen is an important plant nutrient, not just in plants, but in wheat specifically because of its high protein content. Therefore, nitrogen fertilizer is very commonly used. So despite this, the problem lies in the fact that nitrogen is applied in excess to crops, and this gives diminishing returns as the crop yield will eventually flatten off, so you won't get your crops you're trying to make. And so an obvious solution would be to apply exactly the amount of nitrogen you need, but that's not very viable as that's very difficult to assess. Nitrogen fertilizer production also has its own issues because that's harmful to the environment, and buying nitrogen fertilizer is expensive for a lot of reasons. So the aim of the project is to gain a better understanding of how nitrogen responsiveness is controlled and how increasing nitrogen conditions reduce responsiveness in bread wheat specifically. The focus was particularly on this plant hormone called strigalactone, and this hormone has been shown to inhibit shoot branching. And so if we can stop this, we can get more shoots and therefore more crop. So we specifically investigated the strigalactone response and perception pathways. The two hypotheses we investigated were whether strigalactone synthesis responds to the internal or nitrogen status of the plant, and the strigalactone mutants, whether they maintain high tillering phenotype under field conditions. So the first experiment we did was the split root experiment. The purpose of this experiment is the general gist of the experiment is to expose half of the root system to low nitrogen substrate and the other half to high nitrogen substrate. As you can see in the image on the top right, you can see the setup for it. So essentially we had 3 lanes of seedlings, and so the lane on the left and the lane on the right have the roots immersed in only high or low nitrogen, and the one down the middle had half the roots in high and half the roots in low. So this would ideally give us 3 sets of seed plants with different internal nitrogen. So after we grew them for about 2 weeks, we froze them, we cut the roots and shoots apart, and then we froze them so we could extract the RNA, which we then conducted in a technique called qPCR to see how the genes are expressed in these different internal nitrogen status plants. So the idea is that if there's high external nitrogen, the expression should be the same with both their low and high counterparts, but if it responds to internal, it should be similar to each other and different to their high and low status. So here are the qPCR results. So long story short, the first 2 are the biosynthetic genes. So if we look at the first 2, which are D17 and D10A, we can see that the low nitrogen, which is the very far left one, is very high compared to the hatched ones, which are the split ones. So that means that it had to be internal because that's the only way to explain the difference. And if we look at D27 on the other hand, the low nitrogen for both the hatched and the non-hatched show that it's the same, and the same can be seen for the high nitrogen. So that's likely regulated by external. The second row, those are the MAX1 genes, and honestly we have no clue what that's doing over there with the nitrogen regulation. The perception genes are the bottom 2. So the first 2, you can see that the expression of those genes are upregulated by high nitrogen. And because of the low nitrogen, again, the hatched one, it's a lot higher than the one that's lower. You can probably say that D3 and D14 are upregulated at high nitrogen. If you look at D53-4, that one has a high nitrogen bar and a low split low nitrogen bar. So that's likely internal as well. And D53-5, those bars are similar, so that's likely external. So to answer the second hypothesis, we did the field trial. The field trial, we already knew beforehand that the strigalactone perception mutants caused higher tillering in our wheat plants. If you look at the images here, in each image, the very far left is the mutant type, where we stopped strigalactone perception, and the other 2 are the wild types, comparing it to the mutant. And you can see that the mutant types have more shoots than the wild types, regardless of low nitrogen and high nitrogen, but it doesn't seem to really change depending on low or high. But the wild types do have more shoots when it's under high nitrogen. So if we could apply this into a field setting, we can likely do this agriculturally. So there's good evidence for that. So after the field, there's this plot here. And you can probably see already that the mutants are higher there's more tillers already, more branches. And so regardless of the nitrogen level, and the wild types, however, they don't show any, they show little to no difference between the high and low nitrogen conditions. And that's likely an experimental error because during the spring it was very dry, so the fertilizer likely wasn't taken up, resulting in a minimal difference, which is why the one on the far right is almost the exact same, which is not nice. So, to conclude, both hypotheses have been answered. For one, both internal and external nitrogen seem to influence the strigolactone biosynthetic and perception pathways. Specifically, the internal seems to affect more of the genes involved. And when strigolactone signaling is disrupted in the mutants, we see that the more shoots are produced as a result, showing that strigolactone normally helps limit the tiller growth. These conclusions have allowed us to better understand how wheat responds and controls nitrogen conditions. Hopefully these contributions are helpful agriculturally. But before I end, I would like to briefly express my gratitude to the people who made this possible. So firstly, I'd like to thank Mr. Rokos for providing this opportunity, making it possible. I'd also like to thank the members of Girton College who delivered and supported me during my time at NIAB, particularly Dr. Davis and Dr. Swarbrick. And I'd also like to thank Dr. Saha, who was my supervisor and made the time as insightful and enjoyable as possible. Thank you for listening, that's all.